Microbes are most primitive form of life on Earth, and they are present everywhere in Earth's atmosphere. Considering the importance of microbes in research and development, about 750 bioresource centers for long-term preservation and maintenance of microbial diversity are established all over the world. Microbial type culture collection and gene bank called as MTCC, a bioresource center and a national facility, was established in 1986 at CSIR, Institute of Microbial Technology, with the support of CSIR and DBT.
Microbial type culture collection and gene bank, popularly known as MTCC, is the premier microbial research center of this country. It has four major objectives. The one is the conservation and utilization of microbial resources of the country.
MTCC has various divisions taken care by in charge of the sections, and overall activities are coordinated by the head MTCC.
About 12,000 customers who depend on this service provided by the MTCC, be it a school in the remote area of this country to the topmost pharmaceutical and biotech companies of this country, they procure the microbial cultures from us.
MTCC accepts general and safe deposits as well as being an IDA patent deposit under Budapest Treaty. Pure cultures on media plates are mixed with 10% glycerol, and suspension is transferred to cryovials and stored in -70 deep freezer and stored in liquid nitrogen or LN2 containers. Prolonged preservation by freeze drying. Freeze drying is also known as lyophilization, which is a process where water is removed by evaporation or sublimation from the pre-frozen microbial cultures. To prepare freeze-dried ampoules, cells are mixed with sterile 10% skim milk powder solution and filled in glass ampoules, each with 0.2 ml. After pre-freezing, primary drying and secondary drying are performed, and subsequently, ampoules are sealed under vacuum. They are tested for vacuum using a spark tester. After freeze drying, the strain viability and purity is confirmed for each batch of the ampoules. Ampoule labeling is done as per the accession number provided by MTCC, and all labeled ampoules stored in cold room. In October 2002, MTCC was recognized by World Intellectual Property Organization as an international depository authority. Microorganisms can be deposited at MTCC under the Budapest Treaty, and patent protection may be sought in contracting states.
MTCC has state-of-the-art facilities and expertise to characterize microorganisms. We characterize more than 1,000 microorganisms for other researchers in this country every year.
Modern techniques in microbial characterization also used at MTCC. IMTECH 2 is a system works based on the battery of various biochemical reactions like substrate utilization, enzyme production, and resistance to antibiotics, which are characteristics of a bacterial species. The reactions are supplied in the form of a small card where the culture is inoculated and the results are interpreted by a reader through the IMTECH 2 system software, which identifies the culture based on database. The biolog system differentiates microbes based on the pattern of oxidation of carbon substrates. Carbon substrates are presented in dehydrated form in convenient 96 well plates card, where the culture is inoculated and the results are interpreted based on database. The Sherlock microbial identifications system analyzes and identifies microorganisms isolated in pure culture. Fatty acids extracted from unknown microorganism are automatically quantified using GC system and identified by the Sherlock software to determine the fatty acid composition. The MALDI, that is Matrix-Assisted Laser Desorption Ionization. Biotyper identification of bacteria utilizes the protein fingerprint of the cell and relies on high abundance proteins. The mass spectra of proteins expression is compared to reference patterns in database. Molecular characterization is playing a very important role. To perform molecular characterization, a selected gene sequence analysis is performed. To identify fungi, ITS regions are sequenced. The sequences are used for similarity test using database like NCBI Blast Search or Easy Taxon.
Our scientists regularly go for scientific expeditions and explore the areas for new microorganisms. We have so far described more than 100 novel microbial taxa that are new to this science.
Often students from different universities and researchers from various organizations visit MTCC to learn the procedures involved in long-term preservation of microbial cultures and to understand the methods used for the identification of microbial strains.